Her presentation topic is Gauging the Epithelial Mesenchymal Plasticity Spectrum. No? Okay, it's a different topic then. But Professor Joyce is an associate professor and also a Yushan Young Fellow in the Department of Electrical Engineering and Biomedical Electronics and Bioinformatics at this university. And she was formerly the Director of Machine Learning Science at Freenome and a Simons Postdoctoral Fellow at the University of Pennsylvania and UC Berkeley. And she also studied her BS here at NTU and then her PhD at Michigan. And her research focuses on machine learning and computational biology for biomarker discovery, cancer detection, and biomedical data integration. So those groups working with cancer, or if you work with cancer for your own research, please pay special attention to her. And this is a little bit of the guidelines of the report that you have to submit. Please remember that this accounts for 2% of the grade, and the deadline is November 26th. And the grading is based on the top of the content, and then mostly on your perspective and comments. So please give us also your opinion, your very informed opinion, and what do you think you can do with this kind of research. Try to think beyond the talk, but how you can apply this to other areas, for instance. So, and there will be a bonus for asking questions, so remember to participate, and please let me know at the end of the talk if you participated so I can take note of that. So without much further ado, let's welcome Professor Joyce.
Thank you. It's great to see all of you here today. I think I noticed some familiar faces. Hi, this is Joyce. I am a faculty member in the same department in electrical engineering, and I've been in my alma mater here for about 2 years, a little bit more, maybe 2.5 years. What I'm going to share with you today really involves my past research before coming back to NTU. And this is about cancer detection, especially cancer detection from blood. This is some work that I did in a startup company at that time in the Bay Area. That company is called Freenome. And by the time that I left and started my faculty position, we have around 500 employees. So I was the 40th employee at that time, and then by the time that I left, it was about 500. So it really grew rapidly. And why is this important? And how is this kind of blood test for cancer detection is changing this dynamics of cancer screening? So I'd like to share some recent data, not necessarily published by me, but really also show you the growth of this company or the growth of this in general liquid biopsy area. Okay. So at the beginning, let's just start with a little bit of introduction to talk about oncology. Now, it's very broad, but I think it'll be helpful to us to think about what kind of research or what kind of opportunity would you have for AI to apply to these kind of problems. What kind of problems do you have? Okay. And then I will introduce to you blood-based cancer detection. Why is it even possible? What kind of signals can you find from blood? Following that, I'll share with you a few pieces of data that really share with you that machine learning, yes, enables early stage cancer screening, and it is changing the way we screen for at least colorectal cancer. And finally, like some summary. Okay, so first, introduction. Here you see a little paragraph, and I really got this from a New York Times article. The title is a revolution in cancer treatment within reach. Okay, so you all have a screen in front of you. There's a little paragraph. I would like you to take a look, read a paragraph together. Maybe let's also read it together. So in 2014, 18 years after her initial breast cancer treatment, Carol Mackenzie's cancer returned, spreading to lymph nodes in her neck. Doctors do not know exactly why or how, breast cancer can go dormant in a patient's body for so long, not advancing for years, until it suddenly begins to grow. But that's what had happened. Without treatment, Miss Mackenzie's cancer would most likely have made its way to her vital organs and killed her. Over the next 9 years, her oncologist, Dr. Nancy Lin, prescribed 8 different drug regimes, including clinical trials, adjusting treatment as the cancer developed resistance. Some regimes worked for only a few months, while others lasted longer. In 2020, Mackenzie started a new treatment that has successfully stopped the cancer's growth for over 2 years, allowing her to enjoy a near normal quality of life. Okay, so far, a happy ending in this little paragraph. What do you think are some opportunities for AI and ML here just by reading this paragraph? Can we all spend a little time, talk to your teammates, and then later let's discuss with the entire class, what opportunity do you see here for AI? Yeah. Give you 5 minutes. All right, I think time's up. We can continue this discussion after this lecture, but can anyone share with us your thoughts? Maybe one idea from, well, at least almost one idea from each of you. Anything from this table?
We were thinking of predicting which treatment should work.
Okay, which treatment will be effective? Yes, great. Let's go move on to anything from this table. What do you think? What can you do here? Oh, you have a microphone everywhere. Nice.
Like our table thought about the first paragraph and then like some kind of model that predicts when the patient should maybe recheck with the doctor again.
Okay, when to re-examine and
Yeah,
check
making some
for
kind of
cancer
observation
signs.
based on the maybe time series data and then to early recognize the tumor again.
Okay, yes. Cancer detection. And when do you take the test? What about you guys here? Any thought? Okay, what? Predicting reoccurrence. Yes. Yes, reoccurrence. Especially now you already know a baseline for this individual. This patient had cancer before. Did it reoccur? Okay. What about you guys? Yes. So like personalized treatment by reading their genetic report to decide what treatment to give the patient. Yes, great. What about you? All of you here. Any thoughts? Okay. So personalized treatment. Yes, okay. Anything from you guys here?
Micrometastasis or micrometastasis of cells, like identify those using like some FFP images and use those images to identify the micronodes, because that are responsible for the tumor, and that are the cells that are not able to seen by the doctors for a very long time. Like, so maybe identify
Yeah,
those
so
cells.
studying the heterogeneity of the tissue or the medical images that you have to decide when to see the doctor, something like that. Yes. Anything from you?
Yeah, the new treatment maybe can be cut out by generated models.
A new treatment can be found by generative models. So this is about treatment development. So indeed, I just marked a few keywords, but I think there are more that really captures a broad range of research applications, from cancer risk assessment, asking if this patient is likely to have cancer just based on his or her genetics or his or her lifestyle, and cancer detection or cancer reoccurrence detection or treatment selection or treatment development, right? And one key point I want to make is that yes, AIML can make a huge contribution here, but also underlying cancer biology research will also play an important role, and they have to work with each other. So I put it in these 4 topics into the sequence of events broadly for oncology care management, from risk assessment, diagnosis support, treatment selection to treatment development. Okay, so in risk assessment, maybe you will look into this person's personal genetics, right? The lifestyle, smoking status, diet, and diagnosis support, really many different ways. For example, you can take a look at the medical images, x-rays, CTs, MRIs, as well as some liquid biopsy, some specimen that you can analyze, right? So these will be diagnosis support to support the medical staff here to find early signs of signals of cancer. Treatment selection for this patient. What treatment is the right treatment for this patient at this time? So again, many different data modalities. Maybe some of them involve images, some of them may involve even growing an organoid ex vivo, such that you can apply drug to test whether this drug is going to be effective or not. Finally, for some of the problems that just don't have a really good treatment so far, then maybe we have to think about treatment development. And usually some generative AI has been shown really helpful in this area. Okay, so a broad range. Now today, I want to focus more on blood-based cancer detection. Why do we talk about blood-based cancer detection? Well, first of all, let's take a look at some data. In this diagram, you are seeing top 10 cancer types sorted by the rate of cancer deaths in the United States in 2020. Okay, and so the number one cancer ranked is lung cancer, followed by breast cancer, prostate, colon and rectum, sometimes we call them colorectal cancer, pancreatic cancer, liver, so on and so forth. Okay, these are the numbers. Here you can see the top ranked ones. And what's most important is that if you look at the 5-year survival rate of these top ranked cancer types, you see that in this figure there are 3 colors, right? The blue one, the light blue one, represents the survival of those patients if the tumor identified was still localized, followed by a bar next to it, which is in light green. It means that the tumor is still regional. Okay, not too bad. Pretty good survival still. And then the red bar, unfortunately, means those patients that have their tumors started metastasis, so they can find tumors in some distant area, and the survival becomes really low. Okay, so good and bad, unfortunately, the right survival rate is low for some patients, but the good thing is that if you identify cancer early enough, then there's likely some good chance that you will be able to help the patient, okay, and the survival seems a lot higher. So this tells us that early detection is really the key for cancer treatment. And for colorectal cancer, okay, in what follows we're going to talk about colorectal cancer. The current screening rate is still suboptimal. So you can see a table on the right-hand side. This is from a paper that I listed a reference at the bottom anyway. On the right-hand side, you see a few columns. First, it says stool-based, followed by colonoscopy, and then up-to-date. What this is saying is that what is the colorectal cancer screening rate? And stool test is a rather simple one, but unfortunately people don't like to process a stool themselves. Okay, so the detection rate is actually pretty good if you use a stool test, but the adherence rate is pretty low, right? And so overall, only 10% of people who were supposed to have colorectal cancer being screened, so far only 10% follow the guidance. So you can see, see the numbers right here, right? 10%. What about colonoscopy? Colonoscopy is the gold standard. Now, you don't have to do colonoscopy every year. For average risk patient, you have to do it every 10 years. Now, what is the percentage of people that follow this guidance is, okay, 55%. So, either colonoscopy or stool, or maybe some other possibilities that you see listed on the left-hand side in the text. Overall, up to date, only 59% of our average risk individuals or population are following the guidelines to have screening regularly. And this is really suboptimal. Why? Because if we do have the screening rate a lot higher, it means that for some patients, they will have their colorectal cancer identified a lot earlier, which means that they will have much better survival. And at early stages, it's not so hard to treat colorectal cancer. You just have to remove the polyps. Okay. So this is a missed opportunity. This is a great opportunity if we can find a way to help us improve the screening rate for colorectal cancer. And then in the second bullet point on the slide, you see that it's another study. People found that in a randomized controlled trial of individuals who had declined colonoscopy or fit for colorectal cancer screening, offering them a blood test does increase the screening by 7.5%. Okay. And if we provide better guidance, so on and so forth, I think this number can go even higher. So even though the current screening rate is suboptimal, there is a good opportunity for us to develop other type of tests for colorectal cancer such that the screening rate will be higher, such that we can save a lot more lives. Okay, so it's clear. Any questions? Yes?
Did you say cost is a factor?
Cost is a factor for low screening. I don't think so, because they should be covered by insurance.
Maybe
Yeah.
like education or lack of awareness?
Yeah, I think so. Yes, yes. Now, colonoscopy, unfortunately, is probably not so comfortable, even though it's every 10 years. FIT or stool-based test is just that people just don't want to do it. Otherwise, it tends to be pretty cheap. Yes, please.
Do you think these screenings should start at an earlier age? Because I heard, for example, breast cancer is more common that it starts even earlier than that.
Yes, great question. So people did do the analysis previously. That's a great question, by the way. Previously, the screening is required to start at 50, at the age 50. And it was just like in recent years, they updated guidelines such that people should start screening at 45. This is US. Okay. So indeed, it looks like starting earlier is going to help as well. Now, your question is, should we even make it earlier, right? And so that really involves some economic computation, like the risk and benefits, the cost and benefit analysis. Okay, and it's because of those analysis, people change it from 50 to 45. Is it necessary to change, further? I am not sure. I'm not sure, but that kind of analysis would tell us the answer. Okay, so looks like in some recent studies, a blood-based test for colorectal cancer could help. Now, how do we build a blood-based colorectal screening test? Now, the idea is that, well, we should think about the disease progression, okay, how the tumor grows. So in this slide, you see a cartoon diagram. Starting from the left-hand side, we have adenoma, so like pre-cancer state, in situ stage 1, 2, 3, 4, as it progresses from the left towards the right. The thought is that to detect the late-stage cancers, right, we probably can identify some obvious tumor-derived signals and things like ctDNA. What is ctDNA? Circulating tumor DNA. Okay, these are the DNA fragments coming from the tumor cells. Will be very effective. But we also care about early stages of cancer. So if the tumor-derived signal is not as apparent in early stages, then some non-tumor derived signals could be helpful. For example, these signals from the immune system. If that is the case, then it suggests that we need to combine both tumor-derived and non-tumor-derived signals to build a blood-based test. The question is, what can you see from blood? Okay, so this is another cartoon diagram from Wikipedia. As you can see, once you get a blood draw, you will see a blood draw. You will see a tube like this, and then you spin it. You will see the plasma on the top half of this tube, and then some red blood cells and other stuff at the bottom. Now, what we are talking about today mostly are signals coming from the top part, which is the plasma part. In plasma, there are many things that you can find. For example, cell-free DNA. It stands for, cell-free DNA, cfDNA stands for cell-free DNA. It means for those cells that went through apoptosis, okay, when a cell dies, to be using layman language, the DNA in the nucleus will be released into the bloodstream. These should be digested but because there are proteins binding on the DNA, so there are some fragments of DNA tend to exist or survive in a bloodstream for a longer period of time. That gives us a great chance to analyze these little DNA fragments. These are cell-free DNA. Now, if that DNA pieces is from a tumor cell, we call it a ctDNA, circulating tumor DNA. Of course, there may be also cell-free RNAs, cell-free proteins, so on and so forth, and the idea really is to collect those information. Now, each of those pieces of information could involve lots of research. To conduct the wet lab experiment, the molecular biologist will come in and suggest, okay, how do we extract those information from blood? Followed by bioinformaticians to see how do we analyze the data, how do we normalize the data, and then coming with some machine learning models to aggregate all of these different information together. So already from this diagram, you can probably picture in your mind that this is going to be a multimodal model. Okay. Then I said there are many different kinds of signals to look at. Yes. Now, even if just focus on cell-free DNA itself, you already have tons of ways to look at the signals. This is a few references on the slide. Just for cell-free DNA, you can use it to study the mutation from the tumor cells. That will be genetic information. You may also study copy number ordination. This is also at the DNA level. It's genetic. But there are also some epigenetic signals. For example, we can study the methylation patterns. We can also look at the nucleosome positioning. We can also think about chromatin organization, especially given the fact that DNA is not a 1D signal, right? It really has a 3D structure. So all of those can be inferred from blood, from cell-free DNA to some degree. So we can use those, leverage those information to build our blood-based colorectal cancer test. Now, what is cell-free DNA? What is ctDNA? I think this slide will help us to leverage that a little bit. So if you look at the tube of blood on the right-hand side, right, we just said that we're going to focus on plasma. Okay. Now in plasma, you see some pieces of DNA fragments, and they could come from the normal cells. They could also come from the tumor cells. Now, even if they are coming from the tumor cells, and we know that tumor cells have mutation, it's not always the case that you are going to see a fragment with a mutation if that fragment is coming from tumor cells, right? So, if you are going to look for mutation from cell-free DNA, yes, it is possible, but you will be looking for really a needle in a haystack to find that mutation. Okay. But yes, this is one type of signal. If you detect a mutation that is very well known for colorectal cancer, likely then you have detected cancer for this patient. But in order to do that, the chance is pretty low unless you have tons and tons of liters of blood, which is not practical. Okay, but in theory, yes, the signal exists. And so as I said right here, if I am going to screen for a mutation, maybe it is this one right here, right? G is mutated from A. Then what I would do is that I have these pieces of DNA, I align them to the reference genome, the colored matrix on the top, and I search for that mutation. Now, unfortunately, as I said, the fraction of ctDNA that has exactly a mutation on it is very low. On the slide, I said that roughly the probability is 0.01%. Okay, so even just to look for that single mutation, you would need super high coverage, as I listed here. People did some analysis that you need 5,000x, which means that, what does coverage mean? It means that for one base pair, you need to have 5,000 copies of that in order to detect that single mutation. So in theory, yes, the signal exists, but practically it's very hard to find or leverage this as an early cancer screening test. What are something else that we can find? we can study the copy number ordination. So I have a normal sample shown on the top as the reference genome. I have a tumor sample shown on the bottom, okay? And unfortunately, as tumor progresses to stage 3 or stage 4, you start to see the DNA of the tumor getting really messed up. For example, there will be a piece of DNA being inserted. There could also be a piece of DNA that has been deleted, okay, gain and deletion. If you see that behavior, what do you think the cell-free DNA profile will look like? Well, here's one example, okay. Imagine along the reference genome, some DNA pieces are missing, and some DNA pieces you have lots of copies. So if you align them to the reference genome, you start to see a step function for the coverage. On the slide on the right-hand side, people did some simulations by mixing the normal samples with the tumor ones with different proportions, and then you can see as the proportion of the tumor increases, you start to see this step function behavior and the coverage, right? So if you see this kind of signal, like the step function behavior I said, it's probably already pretty apparent that this is stage 3 or stage 4 patient. Now, of course, even just to look at this data, there are also some interesting math questions, right? I can ask, what is the fraction of tumor? Because really, in your cell-free DNA profile, some of them are coming from the normal cells, some of them coming from the tumor cells. It's a mixture. So can I build a model, it's like a mixture model, to solve these parameters together, right? Where is the insertion, where is the deletion? If so, and what is the fraction of tumor versus the normal? So that's copy number ordination. What else? One very important piece of information is the methylation pattern. So we all probably know that our DNA consists of A, T, C, G, these 4 base pairs, these 4 possibilities. For the C positions, you can see the chemistry shown on the top right corner, right? So if you have methylation, then you will have one of the spot now was CH3, right? And it can be unmethylated. So if you look at these C positions, these are called the CpG islands. Essentially, you will have data collected at these CpG islands answering the question, is it methylated or not? So if you collect these CpG islands, you're turning them into a sequence of 0 and 1s. It's a binary signal. It's a binary sequence. Okay. So just like this slide, we're sharing with you what it could look like. And again, tons and tons of ways you can think about how to model this type of data, this sequential data. This paper that I listed is very simple. It's just simply going to compute the fraction of methylation, i.e. the fraction of zeros or the fraction of 1s in this diagram. And that itself seems to be a good biomarker or a good indication of the patient having cancer or not. But of course, you can also start to ask your question, right? What is the pattern of 0 and 1s that has an insight about cancer? I like to say, is 0011 the same as 0101? Well, if you compute the fraction of 0 and 1s, these 2 signals will be the same. But if you look into the patterns, they will be different, right? So how do you model that? Is going to be very interesting. Not only that, the cell-free DNA fragments also tell us the nucleosome positioning. And this has something to do with why do we see the cell-free DNA fragments? Well, it's because the DNA wraps around the nucleosome. As shown on this diagram, you can see the blue curve really represents the DNA that wraps around the gray histones, okay? The histones right there, a collection of them we'll call a nucleosome. Now, if you look at the fragment length and the percent of fragments of that length, which is shown on the right-hand side of the figure, you see that there's a peak around 165 base pairs. Is it random? No, it has some good meaning. It is because In order for a DNA to wrap around a nucleosome, you would need 165 base pairs. So that also explains why do we see these cell-free DNA fragments in blood. It's because these pieces of DNA have nucleosomes binding on them. And the fact that there is a protein binding on the DNA, it protects the DNA from being digested right away. So that gives us a chance to extract those cell-free DNA fragments and study their patterns. And if you align those cell-free DNA fragments to the reference genome, you start to see something really interesting. For example, oops, I always hit the wrong button. Right here, the green coverage, okay, that is essentially by aligning the cell-free DNA fragments to the reference genome, and you see that the coverage has a periodic pattern. And again, this is meaningful. What does it mean? It means that for the DNA to have this 3D structure in the nucleus, there are some specific patterns of the nucleosomes binding on the DNA such that they can form the required structure. Okay. Now, when you look at these signals, probably for some of you in engineering background, you start to think, hey, this looks like time series. Yeah, it's just that the x-axis is not time, but now being location, okay, physical location. So you can also apply many of the techniques for time series modeling to these type of questions to build your predictors for cancer. We also take a look at chromatin accessibility. Now, we have lots and lots of cell-free DNA fragments. We align them to the reference genome. And here we decided to focus on the region around I always hit the right button, to look at the position close to transcription start site, which is coordinate index by 0 right here. What happens there is that in order for a gene to be expressed, right, you have to transcribe the DNA. How is DNA transcribed? Well, you will have the transcription factor coming to bind at the transcription start site, which is abbreviated as TSS at index 0. So if you look at this heat map, this is showing you at each of the genomic position, okay, we all align them to the transcription start site and start to see what is the distribution of the fragment sizes. So you see on the x-axis you have the coordinates, genomic position, whereas on the y-axis you have the fragment length. And of course the color really represents the number of reads of that specific genomic position and of that read length. I'm not sure if you noticed that, but one thing very important is that you start to see many of these fragments around 165 base pair periodically appearing around this TSS site. You also have some brightness right here, which is close to 320 base pairs. If you remember, 165 base pair is the length you need to wrap around a nucleosome. What about 330? Well, maybe there are two nucleosomes right next to each other and the DNA just wrap around them jointly, and so that is called do I have a name? Double nucleosome wrapped fragments, right, on this slide. And you also have these shorter ones right there at TSS. Well, that's around 80 base pairs. The length is 80 base pair. Turns out 80 base pair is the length you would need to wrap around or for a transcription factor to bind to your DNA. So this is interesting. If you do observe the 80 base pair fragments at this transcription start sites, likely that gene is active because it's being transcribed. If you don't see that 80 base pair fragment at the TSS site, maybe that gene is inactive. In that way, you often will just see periodic signals from the 165 base pair nucleosomes. You don't see any signs of the transcription factors. Okay, So by turning our cell-free DNA signals into this 2D histogram, now we have a 2D signals or 2D image for us to study. And we have been building models to predict the gene activation score for genes. Now you may ask, well, what training data do you have? Well, we look for the housekeeping genes. The housekeeping genes means those are the genes that are always on or off in cells. So we can use this as the training data, and they're housekeeping genes, so we know that their label is either 0 or 1. We train a model on these housekeeping genes and apply that model to other genes, and that model will output a score between 0 and 1, which represents the gene activation score, or the probability that that gene is active. So with that, we also have looked into the gene activation score of this patient, and we have this score for all of the genes that we, so on the order of, let's say, 20,000 genes that we studied, and that can also be part of the prediction model for cancer. And interesting, we also compare this gene activation score to the real measurement of protein abundance, and we see some good concordance. And so this is a nice confirmation that the gene activation scores computed from the cell-free DNA fragments, it's really recapitulating the underlying biology. So here on the right-hand side, you see a few diagrams representing the relative protein abundance of stage 1, 2, 3, 4 patients. And this is a very famous biomarker called CEA. As you can see in stage 4, it's been highly elevated. At the bottom, you have also another plot that is showing the gene activation estimates, right? And also you see in stage 4 is highly elevated. So this is, again, a nice concordance that the gene activation score computed from the cell-free DNA fragments is indeed showing us some signals about gene activation, confirmed by the downstream product, which is the protein. Right. Okay. So, any questions? We talked about many different kinds of signals that we can look for in cell-free DNA. That's a lot, and you can come up with even more. Just by looking at these 1D little pieces of fragments, it tells you a lot. Yes, please.
Just out of curiosity, how big is the housekeeping genes data set?
Around 2,000 genes that we looked at the time, yes. Yeah. Okay, if no further questions, what we are going to do next is that plug in all of the information we have and some other things that I just can't share with you, but we start to build our multimodal to predict colorectal cancer, and we have shared these data publicly in a few conferences. For example, we first share our results on colorectal cancer as a preliminary results in 2020 ASCO GI. And we decided, okay, that's great. Let's also look into advanced adenoma, which is a pre-cancer state. So that is published in the next year, 2021 ASCO GI. Not only that, recently this year, we have the model locked and applied to a large clinical trial. So it applied to around 27,000 patients data, a real clinical trial, and that is recently published in 2025 JAMA journal paper. So let's see how it works. Yes?
How routinely collected is cell-free DNA?
Sorry, I didn't hear the question.
How routinely collected is cell-free DNA?
How routinely collected?
Yeah, is it collected at hospitals or only for research purposes?
So far this is for research purposes. Now how can it be collected? You can collect it in a clinic, but you can also just get a blood draw. Well, if it's in the US, you go to like a CVS. It's just like 7-Eleven in Taiwan. Right. So it's very simple. They usually, in the US, they have little pharmacy in those convenience stores.
Oh, so it's actually convenient for
It's actually very convenient because you just need a blood draw. And then once you get the blood draw, the sample will be shipped to, at that time, our headquarters to process it. So we will require them to ship the sample to our headquarters in 2 days, for example, by FedEx.
Oh, so how knowledgeable is the general public about this? Or does a doctor recommend it to their patients?
Not yet, because
Oh,
it's
okay.
not FDA approved yet.
I see.
We have the USC, we have the clinical trial ready, and so it's under review right now by FDA, and we're hoping this will be approved next year. So once FDA approves it, then yes, we can start to have them in the clinics or in convenience stores. Yeah,
Okay.
yeah,
Thank
yeah,
you.
yeah, great question. So this data is a bit dated, but it does shows you the progress of a company or the research team I was involved in, how we went through all the hurdles. This is example number 1, preliminary multiomics results on colorectal cancer detection. As you can see, the sample size is not super huge, but it's already one of the largest at that time among these industry leaders. Okay, so we have 32 CRCs. CRC stands for colorectal cancer. And we also have on the order of 500 colonoscopy confirmed negative subjects. We say colonoscopy confirmed because colonoscopy is the gold standard. We have to use it to confirm that this patient is healthy, at least without colorectal cancer. Okay, and so we have the demographics shared on this slide. And at that time, we apply whole genome sequencing, bisulfite sequencing. Bisulfite sequencing is used to get the methylation signal. We also have protein quantification, and by using cross-validation approaches, here are the preliminary results that we achieved. So how do we read this diagram? Well, on the right-hand side, we have the multi-omics blood test. On the left-hand side, we have using the results using a single protein biomarker called CEA, and that's just very well known. So we compare to that one. The good news is that the bar on the right is so much higher than the ones on the left, because the ones on the right really combines lots of different signals that we can extract from blood to build the test. Okay, and at that time we have shown that we have 94% sensitivity and 94% specificity. Okay, so pretty good. And FYI, there has already been studies, substantial studies, and they have the agreement that as long as any company can build a test that has sensitivity over 74% at 90% specificity, it will be covered by health insurance in the US. So it's good to know that there will already be reimbursement policy existing at that time. So lots and lots of companies are really racing for this. Okay, so if we can achieve 94%, that will be great. But the bar, anyway, is 74% sensitivity. That was colorectal cancer. What about a more earlier state? This is not cancer yet, but a pre-cancer state called colorectal advanced adenomas. Sometimes it's just called them AAs. Okay, but we decided to apply a similar approach to this set of samples and see how well we can do. So we have here around 122 AA samples, and we have 400-ish negative or healthy individuals. And we decided to look into proteins along with methylation signals, aggregate them together with our machine learning model. And the results is shared on this slide. As you can see, again, the multi-omics test is on the very left-hand bar. And it's higher than looking at methylation alone. It's also higher than looking at CEA, that famous biomarker alone. So this is great news to us. It again confirms that if you can leverage multi-omics signals and combine them in an appropriate way with machine learning, you should be able to achieve pretty good performance for detecting cancer early, at least colorectal cancer early. So these two examples, example number 1 and example number 2, really are preliminary results during discovery or during our research R&D phase. If you look into multi-omics, some of them are for discovery purposes. It's very hard to design a protein measurement for all of the ones that we use in this study. So for production, for our real test, at least for the first version, we only select a subset of those proteins that we identified. Okay. Now, how do you select that protein list is also a very tedious piece of work for you to conduct in your research. And so we did that, and we locked our model, and we applied to a large cohort. This is the largest colorectal cancer clinical validation set of samples in the world so far. So starting from 2020 up to 2022, we have collected, how many? 48,000 patients. That's a lot of money. And also because unfortunately at that time it was COVID, so the sample collection process, the protocol changed a bit. So in the end, we decided to set aside a bunch of samples, as you can see right here, around 16,000 of them. We just don't touch them. And then we use the remaining 32,000 samples for the clinical trials. To evaluate the test that we have locked. And there will be some exclusion criteria, like withdrawal by participants, or excluded due to colonoscopy or histology problems. So as you can see, a clinical trial is very hard to run. Even if you collect a decent amount of them, these samples you just can't use. So from these 32,000 patients, in the end, we use 27,000 of them as evaluable participants, and we apply our test to these individuals. Okay, and so here is the results. So overall, if we don't consider the stage, the colorectal cancer, the sensitivity is 79%. Okay, and the specificity, as you can see, like the negative findings right there, I think it is, we intentionally set the threshold such that we have specificity higher than the bar. So here we have 91.5% as the specificity. So, okay, we passed the bar. What was the bar? The bar was you have to achieve 74% sensitivity at 90% specificity. So we have something higher than that. Of course, if you break it down by stages, right, you can see 1, 2, 3, 4, the performance really varies, and the one that we still need to work a lot harder is for stage 1. And if we look into advanced precancerous lesions, it's only 12.5% in its sensitivity, of course, something much lower than what we just saw in example number 2. But this also tells you how difficult it is if you have such a small set of samples for training purposes, for research purposes. If you apply it to a large, huge cohort, generalizability is very, very challenging. Okay. The good news is that we pass. And not only that, if you look into the distribution in terms of the age and sex in this clinical trial, it's not exactly matching the US Census. So we look into the US Census data and try to adjust the weights accordingly. And with that, you can see in this table, the observed, what we said was 79% sensitivity. If we adjust by the US Census, age-sex distribution is a little bit higher, 81% sensitivity. Okay, so we did pretty well, and people are happy that, and this is the very last slide to share with you. So Freenome is a company that I worked at, so all of the data I share with you are these publicly available data sets shared by this company called Freenome. And recently, Freenome and another company called Exact Science has an agreement. Exact Science is the company that is doing really well. It's building this stool-based test that we see at the beginning. But unfortunately, the adherence rate for screening is pretty low. And they also started to build their blood test, but they are not doing so well in terms of performance. So they decided to license with Freenome to have the exclusive licensing agreement, which means that they're going to sell our product. And so this is the news released by these two companies recently in August this year. It's a long journey. I've been there for almost 6 years, and the recent clinical trial data was already after that I the time when I leave the company. And in order to get these tests really done and be available to patients, you can see how long it took. This company has been around for more than 10 years. Okay, it's a long journey, but I think it's a worthwhile journey because it is going to make a huge impact on patients. And for many of you here, interested in AI, also interested in healthcare, this is a good example for you to think about what kind of problem do you want to solve. And hopefully with your training here, you have a good set of tools, equipped it with you, so you can go ahead to think about what is your problem you want to solve and then apply these set of tools to that to make a huge impact. I think this experience for me is really special in the case that I think these impact a lot more meaningful than the ones if I worked in an IT company, right? Because this is directly impacting patients' life. So that's pretty much what I have for you today. Very last slide. Coming back to the beginning, we talked about oncology care management. We talked about some possibilities. Well, you bring up many different possibilities, right? Motivated by that little paragraph. So not only machine learning, but cancer biology, all working side by side. I think we can make great progress in many different areas. This is not an exhaustive list, but a good list of categories for you to think about. All right? Okay, I'll stop here. It's about 1 hour. And we are supposed to have discussion after this. Do you want to have a little break before discussion, or should we just jump into it? What do you say? Right sounds good. Okay, so then we'll have 10-minute break, right, usually. Okay, so it's 15:16, so we'll be back around 3:26.
adenoma，对啊，因为它是 pre-cancer， 所以它更难啊。
对，它的背后的关键是因为 cancer 的这些 mechanism 的关系，
对，
还是说是不是data collection？
那个根据我的经验以及看别人的研究，本来就是 pre-cancer 就是很难 detect 嘛。这个应该也符合预期，就是 stage 1就比 stage 4还要难 detect 嘛，那它是比 stage 1还要更早的。对。
因为它的发展过程，
它的前面，或者是说前面它还没有什么不正常的 signal release 出来，对不对？
所以它资讯还不够，是比较容易被 noise 干掉，以至于 model 出不来这样子。
对啊，而且就没有什么 biomarker 是 highly associated with pre-cancer stage。如果有的话，那就很容易做了。
因为就是还是想说尽量把侦测目标放到越前面，
当然，
这个拉越高，应该是越小众。对，如果说要针对这个部分做improvement的话，应该会朝什么方向？
我们提出来的就是希望可以看 multi-omics， 因为可能不同的 biomarker 除了不同的 signal， 针对不同的 stage of cancer， 它的那个关联性不一样嘛，也许每 stage 比较什么，对，所以
已经是用 multi-omics，
已经用 multi-omics 了，对啊，那也许是不是要又结合不同的方式，我不知道，这很难。
想说 multi-omics 这已经是配合 machine learning 可以取得最多的资料。
光是用 multi-omics 都已经很难了，因为有些东西你觉得它可以拿来量测，但是呢，它可能就没有那么 robust。更何况你要 develop 的是一个 product， 对不对？所以我今天抽了一管血，我还不见得能够马上去 process 它。那在这个运送过程中，都可能会有 degradation process。很难，但你可以试一试，做得出来的话 ，impact 会很高。
是等于收了这个东西之后，所以它的是只有选，然后一些 self-read 的那个所以就是你面检测的这些，比如说 operation 啊，就是这个设计上就是看，比如说可能是公司的经理，就是你在检测上，他们里面有哪些差别？
我不能说我们最后到底是用哪个，所以我刚刚 share data 全部都是 publicly 其他 researcher published 的一些 papers， 但是只是说 OK， 光是看 self-read data 就有这么多东西可以看，那到底哪一个最重要，怎么样变成一个 product， 就不是我现在要讲的事情了。
那就是因为你的 goal 应该是因为你刚刚提到你的那个 prevalence， 到幼稚
对，
会去愿意接受那个，因为我们就是做那个负面呈现，对啊，所以其实比例不高，其实跟 breast cancer 那个乳色的那个 prevalence 在台湾大概也只有10%，就是大家想要去做的所以现在做这个目的才会变得比较简单，大家都抽血，所以那它的 cost 就是如果我们要去抽一次，比如说我今天我不想做。大概会是多少？
我不知道，我们公司的价钱是什么我不知道，但其实有另外一个 competitor 叫做 Garden Health， 他们应该有一件 product， 他卖多少钱？好像是5万块在台湾，台币。
但是如果你的 screen test 为够的话，就
对，我知道，到底贵不贵这件事情是每个国家不一样，见仁见智。但是我觉得在台湾。如果你要比较台湾跟美国的话，台湾的用那个前血反应的比例还比较高一点，endurance是比较高的，所以大概不会想要抽血。但是在美国的话，可能那个会愿意做前血反应的没那么高，而且反正是他们的 health， 对啊，我刚才已经讲超过一个比例的 performance 的话，健保会付嘛，私家健保，他们的保险都是付，所以对，所以谁付钱这件事情很重要，要有人付钱。但是台湾没有人会给付，保险不会给付这个，所以它变成是你自费，5万块就这样贵的。
那这个方法就是拉分一下，说这个应该像 liquid biopsy 的话，这个算是用 NGS 吗？还是它是用 microarray 还是
？NGS sequence。
那另外是我自己在做一个，就是也是考虑到这个刚刚讲到 market， 我就想到说这个在讲到，因为在做的时候跑马桶，然后它的什么 difference 因为我们就担心是伪阳性，不希望伪阴性，就我们不希望说我跟你说没有，结果你最后去做大，会发现有，所以希望那个伪阴性不要太高，所以我看 sensitivity 都会尽量拉高一点。
我们是这样看的，我们先定了一个构数，我们的 specificity 至少要多少？
讲错了，对 ，specificity 要高一点，对，应该是
两个都希望高一点，但是我们先定了一个 specificity 是哪里，然后就大家都比较好，那这同样的 specificity 加 sensitivity 的比较。
但是就像我在
你觉得要调更高是不是？
我在做 breast 的时候，有时候就是因为假设我们找一个 pathology 是一个病理科的医师去比较，所以我的意思它几乎100%，它的 specificity 几乎是100%，就是因为它直接肉眼看的是多片上。那我们在做的时候，就是单纯用 model 想要去 train 好的 model 要去读好的 data。那后来就觉得，怎么样都很难比上一针的。那所以刚刚就讲的是 model， 所以就是把比如说拉进一起做综合的讨论，
可以啊，对啊，
这个算是也算是 model。
omics 通常指的是那个字的结尾是 omics，proteomics 后面是有个字会叫 omics，genomics 会有，所以各种不同的 omics 叫做 multi-omics， 所以你加其他的东西叫 multi-omics， 但是你可以讲它是 multimodal 啊。因为传统统计会就是会有好几个 factor， 它就好几个 feature， 它就是跑一个 univariate， 然后再去看说，因为有可能会 challenge 你说，因为你把它进来之后，其实这个 feature 在主导，不是因为你的这个 model 或者你算的这个方式，所以我们有需要再去做，比如那有点像算什么 relation 嘛，还是什么？就是说你必须要。
证明说你拉进来另外一个加入一个 feature，并不是因为被另外一个 feature。
你可以做啊，但是有一个 tricky 的问题是说，有的时候并不是说单一一个 biomarker 好了，它如果看起来有效也许 OK， 但是它如果不是那么有效的 univariate 但是你把它放到那个 machine learning model， 其实很多的不同 variable， 也许它跟别人都是一个 complementary 的角色，对不对？所以它是很重要的。那的确你可以用 maybe ablation study， 就是把它移除了之后，看到你的 performance 会降多少。
因为有可能他单独自己本身是他就加一个，
有可能啊，有可能，可以想象得出来这样的一个，如果是 non-linear behavior。Okay.
All right, let's come back to the class. Now comes our discussion session. I think I received some interesting questions during the break. I might as well just share with you here. So some classmates ask, why is the sensitivity for precancer so low? Yes, unfortunately it's low, and I think this is also expected, right? The earlier you want to detect cancer, the harder the problem is. Now, my guess is that because the tumor-derived signal at that early stage, and it's not even cancer yet, it's just very tiny, so it's very hard to distinguish it from healthy individuals. But of course, a lot more work to be done. We have lots of other kind of omics data to study. It's not exhausted yet, but probably the challenge is not so much computationally, but really the wet lab part. How do you extract those signals from different analytes? Okay, and not only that, as I said, this is building a product. So even if there is a signal that you can find in research, if you collect a sample in a hospital and then process them right away, it doesn't mean that you can do so for a product, because just to imagine, this is a test that is meant to be convenient for individuals, right? So once you go to a clinic or a convenience store to get your blood drawn, you have to at least give like a day or two for the samples to be shipped and processed. So you also have to consider very carefully what can be preserved during the shipping process. Actually, you probably may not imagine this. We have done substantial studies even just to look at how temperature during shifting can impact the results of a test. It's a lot of work. Yeah. So I think maybe there are some other omics data that can tell us some interesting signals about precancerous state, but a lot still needs to be done. Like what kind of robust signal that you can find during the shipping process, so on and so forth. Okay, and another interesting question that I noticed is that about the specificity, well, the trade-off between sensitivity and specificity. How we did these studies usually is that we set a target specificity. For example, we set 90 to be the target specificity. And then since all of the models or tests have the similar specificity, we can go ahead and compare, okay, which one has a better sensitivity. Now, what is the right regime for your specificity? Since there's always been trade-off anyway, I think that really involves lots of conversation, discussion with the clinicians or the clinical practice to consider what is the right range of false alarm can you accept, for example. Okay, now people went through the studies and so we set the target to be 90%. And that is not only us, but really across the industry with many different other partners or competitors. Okay, any other thoughts to discuss? Anything? Is anyone? Oh yes, please.
What are the challenges you faced while, initial phases while pre-processing your data set?
I'll say that again. What are the initial challenges for what?
Yeah, pre-processing the data.
Pre-processing the data. Yes, a lot of challenges. During the development phase of this piece of work, sometimes the molecular biologists process the samples and then they come up with some data for you to study, and then you realize, hey, this looks weird. And so it involves lots of iterative loops to talk to a molecular biologist or our other partners who process the samples. For example, one challenge is that it also comes with many lessons learned. Sometimes the samples or the data just came back to looking weird, and so you have to dig in a lot in the data and realize this just doesn't make sense because maybe you observed Y chromosome data in a sample that is labeled to be female. So when that happens, you know, hey, something went wrong, and so you will set up a QC criteria to make sure that the sample quality is good. There are also other scenarios that we look at the data, we notice some batch effects, but we just do not know where it comes from. We notice that there are a few different clusters of data just look different, and so again, involving in very iterative process to talk to the other departments cross-functionally, right? What happened on which day? What is being processed differently for that batch of samples? And it's through these iterative process to figure out the problems. Yeah. But great question. It's very, very practical.
So like the data set is in the BMP format? It's BMP format or it's like some different format, like the data set?
What format?
It's a BMP format, like
PMP, what is it?
It's yeah.
BAM file. Yeah. Because we run next generation sequencing inside the lab. Yeah. So generate tons and tons of data. So process the BAM files. Yes, please. Finding ways to normalizing is always tricky.
I'm just wondering that Professor mentioned multiple times a threshold of 74% specificity and
90% specificity.
90% specificity and 74% sensitivity for FDA approval. I'm just wondering why is the number?
Why is it 74? Honestly, I can't answer that question. This is just something I know in the field. And someone went through that analysis, and they have this guideline group to say that anyone who can build a test that achieves this performance or above will receive reimbursement.
So it's probably like a governmental group.
Yes,
decision.
yes. So not in my hands.
Okay.
Yeah. Yes, please.
You talked about the screening for the detection of cancer was lowered from 50 to 45 years
Yes.
in USA.
Yes.
And you talked about like the cost risk study. Do you know how they do this? Like what is the
Why changing from 50 to 45?
no,
No.
more about like the cost risk studies, like how it is done.
Oh, how it's done in a cost risk study? Yeah. So, what kind of study can you
Yeah.
do to decide changing that to 45 to start the screening? I do not know the details, to be honest, but I imagine this is just me guessing, right? You have to take a look at the prevalence at different age groups, and if you start your screening at 45, how many lives can you likely save by starting the screening earlier? And in order to start the screening earlier, how much are we going to pay, the economic burden that it would incur? Yeah.
Like, how can you say that save some lives can be this cost?
Yeah, this economic study is very complicated. I really do not know how to do that, but that's probably another major area that requires different expertise. Yeah. Okay. Yes. Sophia.
This is a little bit more on the operational side of things, because you mentioned the company had been going on for 10 years, right?
Yes.
But still it doesn't have FDA approval.
Yes.
So how do you get patients to screen, and how do you keep the company going on without being able to sell the product?
Yeah. It raised a lot of money.
So VC fund?
VC fund, yeah. ABCDE rounds, more than 1 billion, 1.2 billion over the years. And how do you recruit patients? These patients are paid to be in clinical trials. Yeah.
So they're not volunteers?
No. And so they will be paid, and they also get a free colonoscopy. So it means that, or already according to guidance, right, for people above 45, they need to regularly screen for colorectal cancer. So at least like every 10 years, you should do a colonoscopy. So we say that, okay, we can pay for a colonoscopy, but can you get us a blood draw before you do the colonoscopy? So if they consent yes, then they'll be recruited into the clinical trial. Yeah.
And with the colonoscopy, you get the ground truth.
Exactly. This is an interesting question, because there are so many different cancer types to consider for building a blood test. Why colorectal cancer? One reason is that the reimbursement path is already very clear, because someone did the economic study and realized, okay, having a blood test, even if lower, the performance is still beneficial. Number two, it has a very clear golden standard, which is colonoscopy, to compare to. Otherwise, I will also personally be very interested in building a test for pancreatic cancer. We tested that, but the difficulty is that it's very hard to find early stage pancreatic cancer because they are always detected too late. Yes.
In the clinical setting, so the blood test is to replace the cord blood test.
What test are you?
The stool test. The blood test is trying to replace the stool test, right?
I'm not sure if I should say it's going to replace or not, but at least it's complementary, right? Because some people don't like the stool-based test. So at least, hey, there is another option for you, which is the blood test. Now, it's not going to replace the colonoscopy, that's for sure.
Okay. So what if the blood test is positive, then the patient will go for colonoscopy?
Yes.
But what if the colonoscopy is negative? Then
great, you don't have cancer.
Oh.
Right? This is a screening test. This is not a diagnostic test. we are screening for patients that have very high risk of colorectal cancer. If so, please follow up with colonoscopy.
But basically the blood test is trying to test the methylation or the circulating tumor cell or something. So it's probably something wrong in your blood, right? So
Potentially, right? But at least they help us to identify the high-risk individuals. Those individuals that have high risk of having colorectal cancer, right? So if they have high risk and they follow up with colonoscopy, likely they will be able to have their treatment early on.
okay.
Yeah. Now, of course, the specificity is not 100%. So yes, there are going to be some false alarms. Yes. Okay. Well, if no further questions, it's a great pleasure to be here and meet many of you. I think some of you we've seen each other in other classes, so really nice to see you here again. If not, I also enjoy seeing some new faces. Thank you for having me here today, and if you have further questions, want to chat, feel free to reach out to me, even just to write me an email. Great. Thank you.
Someone is coming. Some of them already came during the break. That's really for you know the area pretty well.
How did this happen?
I saw the picture 可不可以去干脆国道旁边
我觉得可以把它的抛在车队冲。去那边。
Where?
Inside of the policy itself, that's really
what I found for the calculation data, they just use the layer.
Oh yeah.
Unless they're all like professional marathon runners or.
I would
You're trying to move it. The other day I was thinking
I think a good question was that
What would be our main target? Like the competitor or
starting to find mitigation when it's offered and face
but if it is not, then 75%.
The question is, should we do
reference your work.
What I've done for the first step is I build the next pencil, which shows chromosome which just predicts the sex. So the model will focus on the gender instead of the sex. Because like hypertension is the male there are 20,000 and then model
create a linear relationship all the model. Linear relationship because there are so many exercises and there are other
I've been thinking for my this first step about
Open for my
like summary. I think it will be a summary. I'll talk about what you yes.
So now we instead keep speaking instead
I can do the presentation.
Well, I think AI is so fantastic. Like, I think even if we can use like other tools, because we are going to predict hypertension, we can even predict further like maybe the market with more hypertension like
but that's another cause like maybe there's
but I think we will have more chance compared to so that's what I was saying, like try to build
that's the question is the question is living here in East Coast human pressure.
Right, since we're all clear, then let's have our perspective, our opinion.
So we need to try
yeah, but maybe she had
I think the question is like the question is try to be that pattern
if we are not having this power, like if we are having like data set or like 85,000 like how many patients maybe our project can have market if we want like when those are there like suppose heart rate suppose diabetes heart or maybe even the method we try to connect all the products. I don't think so, like all the factors water.
Okay.
Maybe all the factors of the membership.
I have some other saying that there is some change in funds.
We are having a big factor of how we are going to build relationship Suppose first 25 per day. So how we are going to check first with all these then the second one another second one for example so you will use CPG and then check with the that will help to because if the score for suppose one is very high and two is for very low, so you can but I don't know what to say this. What to say this. I just know because of the mathematics. Well, I think so most of the people are out I didn't get it like what Chinese had, what is that file is about?
Like the baseline on only clinical data.
Yeah. It's not related to methodology. And so in that data, why did you remove the hypertension at baseline?
I thought it was the main like data legal basically because you mentioned that if someone has hypertension at baseline, she has to have it at but I don't
But if someone is having hypertension at baseline, I don't think it is mandatory if someone is having hypertension because it's a genetic factor, like what I would so genetic factors are like some external factors that causes hypertension, so when the patient is up and when the patient changes his lifestyle, so that genetic factors are also changes, the follow-up data is very long, like 4 to 5 years or even more than the chances of someone is having hypertension baseline and follow-up.
some it would be a very strong because yeah, that's what I have is lifestyle. There are that
Yeah, it's very easy, like we don't change our lifestyle consciously, like unconsciously, but it happens unconsciously. Like currently, suppose I'm studying in NTU, I'm feeling like so much stress, irritation, hypertension, but I'm like somewhere else.
geht da auch mal die halt selbst bei der Baseline, ob man das ausschließt, Attention von Followers, Target. Aber ob man das jetzt nicht etabliert oder nicht.
Target variable distribution before its removal. It's like this. Target removal variable distribution after removal is also same. How is this possible? shape before removing the variable?
I just had to look at it again because the presentation format.
Yeah, that part shape after removing the target variable 1615. Oh yeah, we do.
But the thing is, including this I think validation file could be used in we can use application files for validation. Like some validation site is not our responsible, so we can just discard it. Well, yeah, I don't see. So like some validation sites is not. Yeah, that's what I think. There's some field that's trying to classify validation sites and so in our annotation file there's a column trying to like relation and that's columns is trying to classify the data classes. So we should ask him. So like there's an end short and north short and south short and I think this is like the definition of the CCP site. So like in this you see a CCP site, this CCP site is defined as
Well, I think you can look it up, right? You are talking about CPGI?
higher
Which part of it? I think we are going to use
I think we can refer to and then the one I'm going is CPG Islands the section that's extremely higher so the CPG Islands might be the very important part. So like someone find those CPG islands and now there are CPG islands and most outside and out of this one. Because if we
You have to find it. There's so many pieces there.
because if we remove all this like out of NLS, so the more our further we can like touch it down the outside.
Here, because you said the island is
I cannot find any. So like this section is one here.
and also the section here.
Yeah, should be or
Yeah, we can do it.
so like now that's 500 and yeah, if we have a side open okay, yes. That's why I really sad because it's actually an interesting class that was really bad. He gave us the following because he was sitting here yeah, because you know, so we are supposed to write having that paper like a 90 papers is actually quite something like 20 pages. We have to choose between ignore the new ones, not that great, but I think the one that's great thing. So the first is your observation was that you again, I think what it is like I have change so they have but like I'm also not that sure I wanna I mean yeah I might ask them as well like I was wondering if you and the second one ignored because I don't think I want to interview first the second also introduce the live stream now like I don't want to like you know it's also already having it's obviously just a topic and like I don't want to bother people with like asking these questions, you know. But like we still want to just so I mean yeah it's I would be amazing but no it's that's actually easier thing. I was wondering before your recent time especially you remember any like the inside and white or something where you say yeah but really this is or yeah and then you guys always like hackers and like I already like I wanted to join it to they were like in my memory right so I was like oh yeah if there's like I also did some research like there were some bad incidents happening like for example I read about this there was some sex incident I don't know if it was all one I don't know if you've heard after 2 years of things like I need to check that they make and I think
that's why you're so good at that.
that doesn't make any sense. That's like all your Chinese is
it's like nonsense. She's a doctor, so
I don't like parts of necessary
I don't know, maybe the better at that.
Why that's
Which is not as stereotype.
and then the second stereotype is
Yeah. And also
that
like
is wrong in so many.
No, not anymore, not anymore. I long time ago. I did try out. I'm also very good at sports. Swimming is a very
I'm swimmer as well.
Like we are trying to treat them like nothing. But we cannot consider all of them. But I think it's
So you're so great at Oh, that's such a weird one. Like it's weird. Yeah, excuse me, I'm actually said that stereotype of Western sounds really bad. But like what he says, it's not. They're like maybe towards different paths in the models and yeah, I don't know either. I don't really think it's it's really bad. Everything is not right. It's so interesting. You could have made so much more of this. But yeah,
what's your favorite?
yeah. Okay, so I'm saying in general, Germany is still a bit I'd say like so like especially big cities in Germany, like it's easy. It's not an issue. But then again, what I always want to emphasize, I come from like a man. Obviously, I can't really you what I do with my friends, but I've never experienced that. But like what my friends told me, and what I've heard is that like where I'm from Hamburg. But I also very amazed by how big this is. Yeah, amazing. So it's everywhere. So There's obviously no way they want to they definitely against safety. So they're trying I think in that sense they are they want to make the There's South America and that's Taiwan. There's not many Oh, okay. Wow, amazing. You met her too.
There are so many movies. The movie is next time.
So boring. Even in and it's such
and in Germany, we have there are three mainstream heroes. One are one is very famous and another is
it's still also very expressive and romantic
crazy CG effect. But I think France love them so much they also gave them love to this but that's why he's very close to girl.
There are a lot of expression or other types of videos that
Yeah, it's kind of the soul.
I think because as I said, I also I hate this Taiwan and I know because it's very little. And I'll ask because that's
but the association was just not really that changing expression. It's just like a
that would be basic, but otherwise I will find something. I can of course always ask. It's actually but I feel bad, I know, but the it's like no one just make up with it. A lot of time, right? Just very early. But I it's not early because I don't know. So I don't
So I suppose
My strategy was
they're kind of related.
every time like
yeah, so I don't like because
Yeah, same. I wanna like I wanna isolate my space with
two different data.
Yeah, I will try. I will upload that one. You can also send this to you via mail. That's great. Yeah, it's always a German. And then add, I guess, what I'm not sure yet. Okay, nice. Yeah, I will. Yeah, so my idea was just to keep education. But I like that was actually very interesting. Basically starting from the baseline area, like under with the giant trees, environmental changes. Very interesting. Very good. Looking at our data as well, I think yeah, these are very, very easy. It's like I just go there and this and like sometimes you have to apply like but then I'm like yeah, the first way I do. Also it's nice to quality so yeah, if you have questions, I'm okay, thank you so much. That's alright, yeah, we can cut it down to about 5K. We didn't check, but I think I found that but then yours have marriage is planned by most of the yeah, I heard that as well. I also love that. Yeah, I wasn't surprised when I heard this time. Because it was very early. It's sort of the other way around. The society supported it all along, and politics was very late. Even if we yeah, because I was the other way around. Like when the almost passed, and they were showing that all these things were 5% supported. So like it was basically a bit early in the society supported. That means like the other way around. Like the best way to definitely didn't show that the bar is definitely see no, when they run, yeah, that's true. But then like I was strong to make more than
thousands, so
I've been a problem.
Okay, that is cute. What did you somehow? I don't know whether it's late or early, honestly, because I mean, that's Christmas today. But I wonder, yeah, maybe I didn't get important. Maybe once it's like, that would yeah.
Your father, because you had a I mean,
How they can be Saturday, that's why
like, okay, you were telling me, but your mom
I'd love to. Yeah, if you show us a chance to do more information, also to be very excited. Always the other way around. I expert my against the ad as well.
Yeah, in my case, it was like that. I mean, my dad
These two, like, if we have to focus through regulation or everything.
when I was looking at first, I was like, okay, I accept it, but I'll have I'll need some time to
To be honest, I think we should
because my parents have this idea of
yeah,
but that might not much.
we are included in the
Like,
genetic
for some time, you have a girlfriend, and of course that's
it has a shared
but when
effect,
I had a friend who actually so it's like, I'm not sure I'm consider it's words you don't watch or what you actually and now I'm like,
Chinese, like, RS, SMP, yeah, that was in genetics.
The model will learn. 现在大家都找的，
Yeah, that's so
接的单位，因为他们知道是在介绍这个对我们他有什么样的这个宇宙是要看就是有可信度之类的，但是有要对不对？是不是先抓当主干，然后我在这个主干里面实际上可以加入其他的那一个宇宙的东西，其他根本也全部都是只要我们主干的就好，然后我们后续的那个一起的然后再跟我们怎么做，我们是不是可以对那边可以加某几个，但前面就里面的主干为主，然后可能我们再加一点模型在里面。
Yeah, there's a bad way. Anything that goes to bad and accounting or teachers are difficult.
但是我就是不用
我觉得应该没有那么多时间可以介绍。对，所以你们就是有一篇是主干，然后对，
只要是其他题目有主干，它的宇宙就是，
但是你要记得你要按照上面的规矩去
就是一步一步。
对对对，作者你也要去吧，有吧有，它上面有。
主干要秀，那是一定要的，因为我们之前每一步都要这样，这些东西都是主干。
我觉得你
但是就最后I will just 引用自己的然后原本就不用举例，因为主要不在 所以我们就在想，
我是申请那个，我觉得申请上，没有，我们跟他申请后来，我现在又要重新申请，没有，其他组只有那个他们可以选择，他们一进去还在这，我觉得没问题，因为一进去的肯定要那种但是这3天之前我们要先完成那个什么 CITI 的一个课程，老师也要完成，我们课程对，你们也要。
那个连接是这个已经关上了。
到时候再做。还有礼拜5的时候会跟你们讲，我是还因为怎么样？然后
我说我们录完一点。
对啊，因为我有去申请，然后结果他一大堆东西。你看，这是老师的部分，他说这是老师的部分，
这门课
每年都不一样，这就是
因为资料集没办法先传，只有我们有哪些信息？
就这3个是新的。资料集吗？也有，上学期。那个是应该还好。不会啦，我觉得还好，还可以。对，就是尽量，因为他这边我有查过，说这些都缴交的话，他大概3到5天。对，所以但是老师有回我，他叫我碰上面，他今天坐飞机的时候，对他有。
希望那些是。
我要跟他讲说，我可以帮他上课了，你知道吗？你都没有回我，我怎么办？我也很紧张。
但我们就都那个数据的。
就改他的架构，
就我们架构部分就是我们那我们是不是对于这样的我是想说，这种意思。
我的想法应该会是说，要介绍这一边，然后这一边的话，它出来前面是好的吗？很好，因为很好。来看完。因为就是如果是新的，因为这个算是新的学生，意思就是说他的问题就是他有些问题对他对，然后现在找另外一边，另外一边去解决这个事情。
So you don't have to go back, you just have to go out.
But even he don't it's just 1 hour before he needs to go to the immigration.
我觉得可以不用，我们可以说我帮一起找在这，就说他这边处理方式可能会有什么，也不一定是真的太多。
Yeah, last year I was here. I usually go to the immigration office.
那要不然可能会一起来，或者看我们的那个，就是可能都是最后我讲的。你们
你可以讲说，这边里面的话，它只有A B报告，然后但是，对，就把它再过去，找到一个A报告，但主干的加上 A报告还在。
So you are going to Manila?
一定要存在。挑战，我就是想，我就是觉得这些东西都应该跟挑战讲，我们把它讲完了，就没人讲。所以我觉得你们的这个什么一步一步的东西，反而可以让我们最后的时候一起去来决定。哎，你部分是什么？你问一下。 会写。对啊，那你就主干的为什么？
那我觉得这样子好像就是你前面就是按照这个然后就讲说这条，然后就要讲会讲
我的想法是，模型的只是性功力、性能力，这两个种就是对你的来说，这样就引来了想讲的几个字，因为他要讲的还有那个完整的一个情绪的过程，对啊，所以等于是我这个现在只讲的，所以我不会讲太多的事，对啊，不然那种我要怎么做，
应该是前面的背景那些。
所以我觉得你的东西是完全覆盖在就是那一个任务上。
因为你有参考书啊，那些但这会是一个讨论，是因为就是我觉得那个会有一个，就是在研究的时候，会有当就是会想说，哎，我觉得我要什么，就是会有架构，但这些架构是balance 在应该是说在它的在就是沿参考对不对？那这个是参考上，但是我们的 table one table two 会
所以我们的这个主干，它最几乎跟我们要做的性高度相关。这样子才能维持这样子。这是那个第二个 conclusion 这个东西就是讲在后面。
Crazy because last time we were doing all those they were so stressful, so we have to do it tomorrow.
I 觉得以报告来做法，还有另一种针对开始的时候，就是以我全部的直接的，就是做这个，这个很难哦。我在想，这东西10分钟讲一个，是不是你要？
但是你提的话，
对吗？
就是这样比较接地气的话，稍微快速讲一下，它的概念会是。那这个就会是解决方式。
It's probably everywhere. That's good.
就看你们是要把它抄在里面，就是教授在放在最后讲，如果他最后讲的话，
If you even if you open long enough, it becomes regular, but it's not that hard. No, but this is you will love this. This is very good. Why did your parents get divorced? Makes sense.
啊，你还没看完，你还没看完，我很想知道说，
我需要讲16个
I'm available on Saturday and Sunday, so
就那个属于的，
那个第一部分介绍 paper 跟它对我们有什么帮助，只要提主干就好了。所以你们不用讲到其他问题。
所以前面不要花太多。
对，你们不用那么难。你可以专注讲那个自然概念的。
但是目前其他的。
I would be on the campus anyway. Right, let's see you tomorrow. Bye. You don't have to do anything.
这个是 conference，所以它不是那个。
会议请来。
So I should somehow every time I have this class, my laptop is just there.
我觉得这都是机会在这里。
I don't know how I do it. Right.
我觉得这个部分要跟分享。我有点担忧他所谓的介绍3到4篇的 paper 的讲座，我感觉他应该是要介绍第一篇 paper 用的是什么去能力，然后他要因此去转出，但我们想要往主攻的方向，于是我们找了一篇主攻，然后基于这个主攻，我们又想到资料有进展跟动态，于是我们找这个 fusion model 的方式，可以做这样的。
这我觉得你是可以再做一张 slide， 就是写说你是哪一个。
在最后还是这样。主战公是另一个，下列三那个，
这也是 conference。
主战公是下列三那个 AI 专门的第一篇。我说主干嘛，就是我们的主角，那个是我们主要的主攻的角，那不是我们的主干。对，就是我们主要的报告看的。
Just take it. It's a good summary.
对啊，但确认了之后，我觉得就是第一篇，而且最有效应该还是。主要是我们其实找了很多篇，应该办的。
因为 Frontier 的，因为他有一点，
我有点担忧的就是，我现在有两种情况，一个是我可以找到一个或者是我找到跟主题完全不相符，只有模型没用，可是它的内容或者是里面下面的，我就有能力在里面拿成东西。但它甚至是会议请到说出也太适合当做一些。对，大部分就是选择我现在这样。
可能是要有发表。
对。
那 Frontier 有一点，它有一点是那个大部分是
然后我们其他找全部都是那个当主的，所以我们就是CRBS 想要针对但实际上我们那些，那我们早就用那个去可以啊，那我觉得我们后面那几篇找一篇有几个是，
我觉得可以啊，因为你只是把它要参考，就是说它针对的东西反而是
对，那这样子那边我不知道你们是谁负责那个，他对我们那边就讲得清楚，就他对我们有帮助的点是来自于他的什么，而不是他的什么。对，不是他没有什么他的方法，但是他处理资料之后，可能换这边有看法，至少就有在看，在这个不是有没有3 T P 他也是有听的。
You can only withdraw one and theoretically it's actually out of but you have to file I think
还是我们先挑一篇会议不是会议纪要，就是有发表的文章，然后下午的话，
我不知道老师能吗？我觉得
因为开发因为我发到什么就是说你有几篇是当时要的。
Maybe it's lucky it's 4 years. It's not all requirements correct paper. Maybe it's not what I planned with.
depends on how the tutoring paper will get back in.
I think I was really
And for my experience, the paper, I think one paper will cost like 3 years. Yeah, 4 years, or like I submitted, and there's a rebuttal, and maybe a second rebuttal, and they will decide to reject our site. So if after the second rebuttal and the paper just rejected, then I need 1 more years.
So you are going to perform
Maybe next year, like January. But I'm not sure if that paper I will be the first author or not.
Also, you need to be first author.
Yeah, 2 more first authors. Because that paper has a lot of collaborators. Like the members in Academia Sinica and also my lab mates. So I think that is more like a methodology paper instead of a method learning paper. So I don't think I will be the first author. Maybe just a professor.
So are you?
I've already had 1 paper as a first author, but the master. Yeah. So it's like a great my master.
Are you planning for like some
maybe Singapore. There's an A-STAR project. It's for the PhD student exchange, and you can contact their professor, and I think it's their lab will sponsor. So the sponsor will not be NTU, will be I think it's a research center in Singapore.
So it will be more money.
Yeah, way more money. I think maybe like 3,000 or 4,000 in Singapore dollars, so maybe 60,000 in NTUs. But that project need to contact the professor directly, so I need to have something to do.
So like you are going to work for NTU project as well?
Yeah, if I do. Yeah, and also for that meeting is I think our meeting is really free, so I can just attend online. Like yesterday because of typhoon, although there are not really bad weather, but all of the meeting was online.
Professor?
Yeah, professor.
I think so. He is very knowledgeable.
Let's go.
没有啊，因为他们拿的也是韩国的。对啊，他应该不是拿我们对他这个，他应该是拿我们家的。
there's this like hybrid relevance.
它就结合哦。那我们可以结构。
我们可以引用其他的美国去介绍。
你做他的头。
我觉得结构完全可以介绍其他的，但整个
但是你可以说你
可以做吗？
Yeah, but I'm working on it. I have no idea.
所以他这边的 test。
Because yeah, improve the that's like
对，我觉得这样蛮好的。
so it's like really
你们有看到我的地方是吗？都是红色的。
that's probably
然后 paper 也可以。
you have to use a web hack.
一定要的啊。
You have to use a web hack. Yeah, you need for questions. Yeah, for questions really. I also had
我就不要投哪里。哦，这个应该比较难的。可以查一下，我们有一个 list， 可以给我看，然后你可以可以，因为没差。他们那一组最后留下来好像只有一个，所以只有那一位其他的通通都，应该是说你们可以写对，又不是错，就是你可以写
通常7加9就是刚好剩下人。
我们两个有2个人要毕业，要毕业，
可是有个过分，这个过分的事情是，也希望就是只能读。
那我可以从不同的，
我们可以从不同的，
你可以从不同的，就是
很多不同的计划，
对，不同试试看。
就简单来说，我们越多人去，他不一定每个人都会好。
今天都还在说，之后再说。
应该说我们这只有
但是我觉得预警系统，你现在看预警系统跟预测要不要把导管，
它也没有说
听起来就不太一样。对啊，我觉得这蛮好。